Hello, and welcome to this webinar titled Single-Use and Stainless Steel Bioreactors, Quality Factors for Consideration when Selecting a Suitable System, hosted by Biopharma Asia magazine and presented by Dr. Trevor Deeks and Kenneth Clapp. My name is Sri Patel, and I will be the moderator. Now, before we begin, I would just like to remind our viewers that this event will be hosting a live question and answer session at the end of the presentation. You can submit a question at any time throughout the presentation via the Questions tab, located at the bottom of your screen, and we will go through these at the end of the presentation. Now, please allow me to introduce you to our presenters. Dr. Trevor Deeks has 38 years of experience in pharmaceutical development, manufacturing, quality control, and quality assurance. He is registered as an eligible QP under EC Directive and has also practiced as a registered pharmacist in the UK. He now resides in Maryland and has his own consulting business. He has a PhD in peptide chemistry and has extensive expertise in bioprocessing, aseptic processing, and validation. His experience of a wide range of unique and unusual biotechnology processes, including live vaccines, tissue culture products, and cell-based products. He practiced as a QP from 1983 until 2010 in the UK, and his QP experience includes commercial batch release, investigational medicinal products, and specials. He has established and operated quality systems in compliance with the European Clinical Trials Directive and with global regulatory expectations. He has led scale-up and technology transfer projects for large and small molecules, has managed many projects with contract manufacturing organizations, has undertaken a number of evaluations of new technologies, and has performed due diligence evaluations of such products for acquisition. Kenneth Clapp is a senior manager at GE Healthcare, focusing on applications, technology, and integration. Kenneth holds a bachelor's degree in biology with specialization in subcellular biology. He received a master's degree in biological engineering, focused on biological control systems, mathematical modeling, and instrumentation. Early in his career, he worked as a cell culture technologist, produced antigens and antibodies in large scale, and developed hollow fiber-based bioreactors. Spanning 4 decades, Kenneth has worked in a variety of roles with bioprocess equipment manufacturers, including field service, sales and marketing, applied research and development, quality assurance, automation, and operations management. He's been managing bioprocessing equipment capital projects, including supply oversight, documentation, and ERP integration. I would now like to invite our presenters, Dr. Trevor Deeks and Kenneth Clapp, to join our webinar. Welcome, Dr. Deeks and Ken.
Thank you.
I wanted to thank the audience also today for attending, and I'd like to get started by handing off to Trevor. Trevor?
Okay, well, thank you very much, Ken, and thank you, Sri, for that introduction. I also want to thank Biopharma Asia and GE for inviting me to participate in this presentation. This webinar is going to be focused on biologics processing, or bioprocessing as it's commonly called. It will look at the market and industry drivers for selection of suitable bioprocessing systems, and it will also look at the quality factors. This slide shows our agenda for today. We plan to speak for about 25 minutes each and allow about 20 minutes for questions. By the end of this webinar, we hope to achieve these learning objectives. If we don't cover a particular aspect that you're hoping to hear about, we don't want you to be disappointed, and we can cover it in the questions at the end. And I'll hand over to Ken now for the next few slides.
Excellent. Thank you, Trevor, for that lead-in. Let's start with the trends that we are seeing. Factory productivity and flexibility are at the top of the list of nearly all of our customers. Broader product pipelines and diversification among therapies are behind us. Market access is increasing demand for biotherapeutics around the globe. We are seeing a growing requirement related to integrated process solutions, meaning a shift from equipment or unit operation-centric solutions to a more comprehensive, process-centric solution. There is a demand for new production and purification methods for many of the new emerging therapies. Data transparency and availability is becoming essential to manage a broader supply chain, particularly like those associated with single-use technologies, and also for extracting the most value from the bioprocess itself and achieving operational effectiveness. These drivers boil down to three primary elements. Drivers that address patient and shareholder values, increasing drug success rates, reduction in time to market, and keeping production costs as low as possible without sacrificing quality. What we hear routinely from our biomanufacturing customers is their need for increased throughput, shorter equipment turnaround times, and lower requalification overhead. To that end, this graph depicts the relationship between utilization rate on the x-axis and the associated cost savings on the y-axis. The red bars represent much of what we've been used to in the industry, that is single-use, single-product, dedicated facilities. Single-use technology, including single-use bioreactors, help improve productivity by becoming more flexible and are capable of supporting multi-product manufacturing more easily, which increases those utilization rates. New processing methods, such as process intensification and continuous bioprocessing, have the potential to increase those rates even further. For our discussion today, we want to outline or describe the type of manufacturing scenario we'll be making reference to. It's a manufacturing scale, either clinical or commercial. It's a greenfield facility, not a retrofit. Stainless steel bioreactor capacity to 10,000 liters, and single use to 8,000 liters, comprised of 4 by 2,000 liter single-use bioreactors and conventional downstream. Although we have used this as the representative scenario, much of what we will present will be applicable to other scenarios as well. So please keep your particular scenario in mind as we go through the remainder of this presentation. For me, having been involved in stainless steel equipment, including bioreactors, I find it useful to compare the two technologies side by side as shown here. For our scenario today, the two 5,000 liter stainless steel bioreactors in the left-hand picture and much of their associated piping are being compared to 4 of the single-use 1,000 liter bags and vessels shown at the right. Let's make 3 key comparisons between the bioreactor types. Manufacturing scale stainless steel bioreactors have been and continue to be engineered to order, customized to meet the unique production requirements within dedicated single product facilities. Single-use bioreactors, on the other hand, are comprised of 2 complementary pieces: the hardware, including the vessel or bag holder, and the bag itself. Single-use bioreactors available from the primary suppliers have been built as product platforms. These platforms accommodate changes, particularly in the bag, to address different processes. Change the process, change the bag. Consumables normally associated with stainless steel bioreactors include inlet and exhaust filters, elastomeric tubing sets, and process probes. Likewise, single-use bioreactors employ similar components. However, the quantity and complexity of the single-use assemblies increases substantially as they become the replacements for the vessel and fixed piping of stainless steel bioreactors. Last among the key items, and not to be overlooked, is automation. Automated valves and position indicators are quite common in stainless steel bioreactors. Conversely, single-use bioreactor bags with their ports and tubings rely on manual pinch clamps, which puts more emphasis on the automation platform to guide operators' actions and receive acknowledgement those actions have been taken. Setting the stage for our comparison would not be complete without also mentioning the biomanufacturing baseline. The process, from the upstream raw materials, like the media and the nutrients, through the individual unit operations and process steps to the final bulk product is represented here. The key performance indicators of productivity, yield, quality, and variability, along with their directionalities, are central to efficiency, plant utilization, and commercial success. Two essential data streams are also represented here: workflow and process. Within the context of Industry 4.0, the digital collection and management of this data is essential in making ongoing improvements. Now, back to Trevor.
Thank you, Ken. Yeah, thank you for setting that scenario and the comparison. We'll be getting into more depth about comparing stainless steel and single-use systems later, but first of all, there's a few terms that we may be used during this presentation, and I think we need to define them in order to avoid confusion, since different people may have a different understanding of what these terms mean. So by defining them here, we're all using the same technology, we're all on the same page. The next few slides contain these definitions. If there are any that we've missed here that you would like to have clarified, again, please raise them during the question time. This first slide defines the different types of facilities that we will consider. These three types of facility represent the past, the present, and the future. And this evolution has been facilitated in large part by the advent of single-use systems. Okay, this next slide covers some terms that are frequently used when we talk about contamination control in bioprocessing. I want to particularly look at closed systems. When people talk about closed systems, they're almost always referring to functionally closed systems. And this slide defines what we mean by functionally closed. It means that they're closed when they're in use, but they have to be open to allow materials to be added and for samples to be withdrawn. They also need to be open for cleaning, unless, of course, they're single-use systems, in which case they don't need to be cleaned. It's the way in which opening of closed systems is controlled that attracts a lot of attention from regulatory inspectors. We'll talk a bit about aseptic connections in later slides. Here we define the term single use, and it's important to define this term, even if it seems obvious. The term disposable was used for these systems when they were first introduced, but one regulator, speaking at a PDA conference that I actually attended, pointed out that a system could be used several times before it's disposed of, which defeats the purpose of these systems, and that is not an acceptable practice. So the term single use came about. The important point here is they must not be used more than once. You may have already read about the concerns around extractables and leachables in single-use bags and bioreactors. And here we define both terms and show the difference between them. And the important point here is that extractables are substances that could potentially migrate into the process stream, but not necessarily, won't necessarily in your circumstances. Whether they will or not depends very much on the properties of the process stream, such as pH, solvent content, salt content, and temperature. Leachables, on the other hand, are substances that will migrate into your process under your process conditions. And it's therefore necessary for the process owner to evaluate these leachables under their own process conditions. Vendors can provide a service for investigating leachables if required, but the responsibility here lies with the process owner. I added these slides so that we don't forget the importance of quality. Even the founding fathers appreciated this fact, and it's even more important and relevant when we consider the nature and purpose of the product, the circumstances in which it will be used, and the critical and sensitive nature of the processes used to make the product. We can't afford to ignore quality, and in bioprocessing, there's no reworking, and reprocessing has to be strictly controlled and fully validated. And I'm thinking of things like refiltration and repeating a chromatography step, which are both considered as reprocessing by the regulators. Bottom line, there's very little room for error in bioprocessing. If you make a mistake, it can be costly. For example, if a cell culture gets contaminated, the batch must be discarded. So it's worth taking a look at the historical situation before the explosion of single-use technologies in the bioprocessing business. At the beginning of the millennium, the only single-use systems that were in common use in GMP bioprocessing were disposable filters and bags for buffers. Sometimes these disposable filters were used multiple times, and some examples of that would be air vent filters and sponge filters, which could be used on multiple occasions. 100 litres was considered about the smallest scale for mammalian cell culture for phase 1, and phase 1, of course, is the first point at which you need GMP material. Quite often, the process had to be scaled up for phase 2, and then again for phase 3 and commercial production. By the time you get to phase 3, you should be at your commercial scale. The result was stainless steel bioreactors with many feet of piping, supplying water, gases, and nutrients, supported by extensive media and buffer preparation facilities and storage plants. These facilities were often dedicated because of the regulatory hurdles required to show decontamination and the time taken for product changeover. They were uneconomical, they required a lot of risky capital investment, and at a very early stage of product development. At that point in development, companies don't want to spend that kind of money. You now see a lot of white elephants out there. Companies have these big facilities, dedicated facilities, that are no longer useful at all. Closed systems were difficult to achieve, and most of the aseptic connections were performed in strict clean room conditions, involving strict gowning regimes, airlocks, and personnel flows. So that was the situation. Single-use bioreactors, and particularly single-use systems, and particularly single-use bioreactors, have been a game changer for early phase bioprocessing, and also for later phase. Because of the lower initial cost and the time required to design and construct a facility, companies in very early phase development can manufacture their phase 1 material and get into the clinic much more quickly. Multi-product facilities have become easier to operate, with rapid changeover between batches and between products. Regulators have become more pragmatic about the acceptability of multi-product facilities because of the elimination of the need for cleaning and sterilization, and the elimination of the reduction in validation required, and the improvements in facility decontamination. There has, at last, been a realization that if you can show, using spore strips, that you can fumigate your room and it will penetrate and decontaminate your room, then you have an effective fumigation process. That wasn't always the case, especially for companies handling viral cultures in the same facility as other cultures. Regulators used to ask companies to show that viruses could be recovered from non-product contact surfaces, and that's almost impossible. That attitude has gone away now. And this change is due in part to the fact that single-use systems are closed before they enter the facility, and the aseptic connections are now much easier and safer. In order to make a well-informed decision about system selection, we need to do some very early assessment of our process. We need to know that the process can be run and is scalable in the system we wish to select. Not all cell lines can be propagated or will express protein in single-use systems. So some very early design of experiments is needed, and it's something that most companies don't think about till at least phase 2. They don't want to spend money on design of experiments at phase 1. But the extent of the process understanding needed at this early stage is nowhere near as great, and it is essential for the selection. It's time and money that's well spent, and it can result in a much greater cost and time saving if a single-use system can be used instead of a stainless steel system. So let's look in more detail at quality by design as it applies to the selection of single-use systems. Oh, sorry, rushing ahead. The cell culture process needs to be well controlled to ensure the best yield and the best product quality. It's well accepted that the control of critical process parameters in stainless steel systems is far better and more established in single-use systems. With stainless steel systems, it's quite normal to have more mixing and aeration and temperature control capacity than is needed. More work is required to evaluate single-use system capacity and capability, but the technology is improving. And with the development of jacketed single-use bioreactors and improved agitator design and sparging, it's starting to happen. Either technology will allow pooling of the harvest from multiple bioreactors, and this allows scaling of the downstream process without having to change the upstream process. However, remember, stainless steel reactors require a much larger footprint, and for these systems, it's much more efficient to increase the size of the bioreactor rather than the number. Single-use systems offer almost no opportunity for cross-contamination. This isn't the case with stainless steel systems. In stainless steel systems, there are more opportunities for microbial contamination and inefficient cleaning. These include large piping runs that have bends and dead legs, elastomeric seals to which proteins love to stick, and tanks that have spray ball blind spots. Spray ball coverage is an important criterion for qualification of stainless steel bioreactors and tanks. As we've already discussed, the greatest contamination concern for single-use systems is loss of integrity between leaving the vendor's warehouse and being assembled in the clean room ready for use. Integrity testing in situ is therefore of vital importance. And on that note, I'm going to hand back to Ken.
All right, thank you, Trevor, for that discussion on quality by design. Let me get to, here we go. So this graphic shows the well-recognized drug development path. Introducing quality by design, as Trevor said, early in this journey pays considerable dividends, especially for those specific critical criteria or critical quality attributes of process performance, scale up, material compatibility, and process analytical technology. Making appropriate technology choices helps to achieve a robust bioprocess capable of reaching biomanufacturing scale and matching the objectives necessary there. This is certainly nothing new in the realm of conventional autoclavable or sterilized in place bioreactors. However, with single-use bioreactors, material compatibility and the format of process analytical technology establish additional quality attributes that need to seriously be considered. front and center in the consideration of such quality attributes are details about the process itself. This list of topics represents fundamental process information which is essential to understanding process performance, establishing scalability, evaluating and selecting appropriate technologies. Cell line, terminal volume and scale, existing bioreactor solution, cell density targets, cell growth conditions such as RPM, gas composition, and gas flow rates, if there are any foam issues or CO2 accumulation issues. And although this list may not be exhaustive, it does provide an excellent universal framework upon which to build a body of knowledge necessary to construct bioreactors of either type, stainless steel or single use. Scale-up evaluation can be addressed using process modeling and simulation as well as actual small-scale process runs. The relationship between physical system features such as impeller diameter and type, sparger location and type can be associated with specific performance parameters like power per unit volume, power number, mixing times, and mass transfer capacity. Using bioreactors of the appropriate sizes, the model simulations and relationships can be confirmed with trial runs in process development. These four graphs include results across 3 bioreactor sizes, 10 liters, 2,000 liters, and 200 liters, covering cell viability, cell density, dissolved CO2, and product titer. Results such as this can be used to confirm which critical quality attributes are being properly met across the scales. In these examples, the 200 liter and the 100 liter combine and represent manageable process development sizes to perform this type of confirmatory work. Single-use bioreactor suppliers have expended much effort and expense to create product platforms capable of addressing the broadest possible range of cell culture processes. The bioreactor hardware and the bioreactor bagware must work in concert in order to perform in a way that is analogous to a stainless steel bioreactor. 2 design spaces exist, one for the bag and one for the hardware. The bag design space includes the film, weld-on ports, tubing, agitator drive interfaces, sparging elements, filters, and other similar bag-attached components. The hardware design space includes the vessel or bag holder, process measurement instrumentation, mass flow controllers, pumps for liquid management, exhaust filters, and condensers where applicable, among other things. The bag design space represents the critical process contacting item that can be consistently manufactured to produce a robust, dependable assembly. It establishes the biological compatibility and the mechanical design elements define the sterilizability by gamma irradiation and the physical robustness. The materials of construction must include traceable, documented evidence of material pedigree. In addition to the process requirements already mentioned, user requirements, particularly those associated with bag installation and software interfaces, contribute to the operational success in terms of reduced bag damage, operator guidance, and consistent operation. Facility integration, the attributes associated with utility and site information technology connectivity are also very important considerations. Documentation like test reports, drawings, etc. must be adequate to support GMP operations. The differences between what has been provided with stainless steel bioreactors and what is provided with single-use bioreactors should be well understood. The temptation to copy-paste requirements should be avoided. In the end, the intersection of hardware, bagware, and user requirements determines the true compatibility of single-use bioreactor capabilities to the buyer's needs. In this diagram, the A region defines the intersection of the bioreactor hardware and bioreactor bag design spaces. Region B represents hardware design space with no effect on bagware. And similarly, region C is bag design space having no impact on hardware. The regions labeled D define what from the user requirements is not being met, and understanding this alignment is essential. With stainless steel bioreactors, there's a well-established body of knowledge about the common alloys used and their various forms, such as tubing, valve bodies, sheet material for vessel walls, etc. For such bioreactors, we have relied on metallurgists, and the lexicon has been defined by such terms as corrosion resistance, chromium layers, passivation, and rouging. Supply is ubiquitous, nearly universal, and distributed, establishing independence between bioreactor supplier and buyer. In single use, however, we now talk about plastics and resins, and by analogy, plastic forms that include beads, sheets, extrusions, and moldings. Polymer chemists have taken the place of metallurgists, extractables, leachables, and biocompatibility to find a new lexicon, and unlike stainless steel, single-use security of supply takes center stage because of how sourcing and the supply chain is structured. Single-use assembly suppliers and buyers are linked as never before. Biocompatibility and supply must be considered early in the technology evaluation process. To complete this section on quality by design, we consider probes and sensors. This table is a survey of common measurement types often associated with bioreactors and differentiates between reusable and single-use types. Determining which process measurements are critical to product quality is a fundamental consideration and is well suited to small-scale use. Understanding which measurements are not required at the manufacturing scale is equally important. This is true for both stainless steel and single-use bioreactors. Single-use bioreactors introduce a new probe or sensor form factor, that is, sensors that are welded into the bag and gamma irradiated as part of the bag's manufacturing process. These are ready to use and do not require autoclaving or insertion, unlike their reusable counterparts. In some cases, these weld-in sensors employ sensing techniques that may differ in range, stability, or accuracy from their reusable equivalents. These factors must be considered when deciding which sensor type to use. The image at the upper right shows a reusable probe being inserted into the bag. The lower right-hand image shows a single-use sensor before being mounted to the white bag port plate during the bag manufacturing process. And now, back to Trevor.
Thank you, Ken. I realized while you were talking that I have missed some slides, and I think it's important to just go back and cover those, which I'm doing now. The first one was this slide covering procedures for batch and campaign changeover, and it covers the ergonomics and the logistics of changeover stainless steel as compared to single use. The main difference is that with the changeover of stainless steel, you have to clean and sterilize, whereas there's no cleaning and no sterilization for single use. However, for single use, one of the big considerations is removal of waste, because there's a lot more waste. And that's covered on this next slide. And in order to minimize the risk of cross-contamination due to waste disposal, the waste disposal procedures need to be qualified. You can't really validate them, but you can qualify them and control them. This is becoming the industry norm, and regulatory inspectors will expect it. For stainless steel systems, the validation challenge is generally simpler. The main concern is liquid waste, which is run to a kill tank and then decontaminated by heat treatment. It consumes power, but it's controllable. For single-use systems, the bag needs to be taken out of the clean room and either autoclaved before sending for incineration, or simply bagged up as hazardous waste and incinerated. Autoclaving requires validation, and if the bags are large, with large agitators, the autoclave needs to be large. Removing hazardous waste is much simpler, so simply bagging it up as it is and taking it out the clean room straight to incineration is by far the easiest way to get rid of the waste. However, whether the bag is autoclaved or not, there needs to be a unidirectional flow of materials into the clean room and of waste out of the clean room to avoid the potential for cross-contamination. Unidirectional flow requires a larger clean room footprint, but it's strongly advisable because it's much easier to control and qualify. Leachables from stainless steel systems are not generally considered a risk to product, although stainless steel systems will rouge and passivation is needed during commissioning and periodically thereafter. Main concern for stainless steel systems is clean in place reagents. There's also a risk that these can cause inhibition of microbial cell cultures. Therefore, the validation of the CIP cycle has to also demonstrate that the rinsing will remove cleaning reagent residues. For single-use systems, we need to understand what is going to leach into the process stream. And as has already been mentioned, this is a job for the process owner. We need to understand what the impact of those leachables might be on cell propagation and protein expression. At this stage, we are far less concerned about the leachables getting into the final product. But during downstream processing, we need to understand the toxicity risk of the leachables if they do carry over into the final product. And we also need to understand if they will interact with the product and impact product stability. All of this is part of the early phase design of experiments that we referred to earlier, and it's essential prior to selection. We'll go back now to where we should have been when Ken finished speaking. Oh, here we go. So now let's compare some of the business considerations between stainless steel and single-use systems. Cost is always important, and the comparison here is between high capital costs for equipment and utilities, qualification and validation of stainless steel systems, and the high cost of the usage of utilities as compared to the high revenue for single-use disposable systems. The bags themselves are expensive. There are many cost comparisons in the literature. They include comparison of bag changeover times and other factors, but none of them that I'm aware of has included a comparison of speed to market, and that is a very important cost consideration. Time is money. You can get to your first GMP batch and scale up far more quickly using single-use systems. You can save valuable time when developing new products. The smallest footprint of the single-use systems also provides significant cost benefit. Single-use systems are clearly more flexible, primarily because you can run multiple types of cell lines and scale up all in the same suite on a campaign basis. So you don't need as many facilities or as large a facility. So we've talked about quality by design, but this slide provides a high-level comparison of the quality considerations for single-use and stainless steel systems. Bioreactor integrity is one of the most important quality factors, and the methodology for demonstrating bioreactor integrity in stainless steel bioreactors is far better established. We can assign very tight acceptance criteria for pressure hold tests. If the bioreactor is well sealed, it can hold a high pressure for a long period of time. Bags cannot be pressurized to the same extent as stainless steel tanks, and the sensitivity of the pressure decay test is limited. Pressure losses do occur over time, even with intact systems, and each system configuration needs to be evaluated to determine what the acceptance criteria should be. Vendors are responding to customer and regulatory demands for better defined methods, and there are some custom solutions out there, and the sensitivity is improving. This is something that you should discuss with the vendors and see what they have to offer. There's also a guidance document on single-use leak testing, and it's currently being developed by the Bioprocess Systems Alliance, which is an interesting body worth looking at. We've already touched upon product changeover because of the elimination of CIP, SIP between batches when using single-use systems. Changeover times have reduced from 3 to 5 days for stainless steel bioreactors to as little as a single shift, so much, much quicker. We've also discussed the issues of product contact and material compatibility. When we're considering mixing, temperature control, mass transfer, it's reasonable to suggest that current stainless steel systems are generally better controlled, but single-use systems are improving all the time. Here are some further additional quality factors. The requirement for cleaning and sterilization comes up as a consideration again and again, and we'll expand on this later. The clean room design is heavily influenced by what needs to go into the clean room, and also by the type of operations that occur in the clean room. The clean room design for single-use systems is much simpler and less extensive, which is why it's quicker and cheaper to construct. The probes and sensors that are used in the stainless steel system are not practical to use in the single-use system. Therefore, probes and sensors need to be specifically designed, and we've already heard about that from Ken. There's less experience with these instruments. They can be calibrated offline in situ, but they are different, and they will have some influence on the process control parameters. You cannot just simply transfer a process from stainless steel to single use or vice versa and expect that you're going to be, the measurements you're going to get are the same. There's an argument here for not changing between single use and stainless steel and just sticking with whatever system you've got and scaling up. Although I would argue that sticking with stainless steel is going to be a more costly option. So that said, I'll hand back to Ken.
Good coverage on facility and business considerations, Trevor. Thank you. I'll continue here with some specific examples. There are a number of technology-based changes associated with the facility, many of which Trevor introduced or discussed just a minute ago. And as this comparative representation depicts, the facility's footprint changes due to changes in bulk fluid management and the reduction or elimination of CIP and SIP requirements. Less obvious, though, are the changes in material, personnel, and waste flows throughout the facility. Specifically related to single-use implementation, materials management, batch record composition, bioreactor integrity testing, as Trevor mentioned, and operation are highlighted here as specific areas to consider. Material management is listed twice, once at the warehouse level, where bulk single-use assemblies are received from the supplier and stored. The second time material management is mentioned relates to pre-manufacturing staging of the assemblies at or near the point of use. Batch records can also be considered as part of material management, since the traceability of single-use components and assemblies is accounted for in the batch record, including rejected or failed assemblies and their replacements or substitutes. For integrity testing, it will be critical to consider the value such testing might add the size of the sample set that might be necessary to establish relevant results, and as well the complexity of measuring temperature-dependent pressure decay in a container with elastic walls. Operational automation considerations, where single-use assemblies are involved, relates to ensuring operational readiness and continuity, particularly where operator intervention is required. Sourcing and procurement business functions act as the middleman between internal stakeholders like manufacturing and quality assurance and the suppliers in the supply chain. Sourcing and procurement must focus on supply security to properly support the internal business stakeholders. The warehouse represents a transient location for single-use components and assemblies. The warehouse space allocation and residence time are dependent on how well supply and consumption rates are aligned and how risks will be mitigated using contingency planning. Contingency plans may include inventory levels, supplier-held inventory, as well as looking for alternative or backup suppliers where applicable. Production planning dictates consumption forecasts, and consumption forecasts influence supplier lead times. Throughout this interdependent interaction, the security of supply must be robust and the chain of traceable material pedigrees must be unbroken. Linking materials in the warehouse to usage in the batch record comes down to how effectively the single-use life cycle is managed in the enterprise resource planning system. To get an idea of the impact single-use products can have on building the batch record, here's an example of a single component. In this case, a filter cartridge sourced from multiple vendors. The component document packages must include the same core content categories, such as material certificates, test results, and drawings. Within these records, however, the layout and presentation of the information may vary from supplier to supplier. Available formats and delivery mediums may include human and machine-readable options. Understanding what the suppliers provide and aligning that with your information management methods is a critical consideration for success. For human-readable items, specific training may be required to avoid misinterpretation of variable content and layout between these types of documents. In all cases, however, linking the records and their information to the component item master within the enterprise resource planning system helps ensure accessibility, consistency, and compliance. Continuing to build the batch record concept a little bit further, here's a practical single-use bioreactor example. This includes components like we saw in the previous slide, plus simple-use assemblies such as tubing kits, used for process connections between bioreactors or to bulk fluid management equipment. All the same comments about component data packages, content, format, and delivery mediums apply here too. In this example, the situation is compounded by quantity and complexity of the assemblies. Dealing with rejected, failed, and substituted components and assemblies at the point of use impacts the manufacturing process tremendously. A proper, thoughtful consideration of contingency planning and the information management associated with this material flow is essential, or is an essential part of the single-use life cycle management. Staging for manufacturing. Warehouses are rarely a good place to unpack and store unprotected single-use components and assemblies. Staging for manufacturing at or near the point of use includes many necessary steps, starting with unpacking. After unpacking, temporary storage can be used to allocate and sequence the assemblies. The inspection step is performed to reduce likelihood of certain visible defects, making it onto the manufacturing floor. Visual inspection criteria should be guided by the supplier's knowledge of the product, specific operator training, and experience gained by the drug maker's operations staff over time. The last step in the sequence is loading the bioreactor bag itself. Here are examples of top-loading bioreactor bag to the right and a waste-level bioreactor loading on the left. Integrity testing is commonplace in stainless steel bioreactors, as Trevor said. It has become an active discussion topic in single-use bioreactors as well. On the left is a typical stainless steel bioreactor piping and instrumentation diagram. On the right, a representative single-use bioreactor P&I diagram. Construction and complexity differences should be clear. Therefore, it will be important to consider the equivalent effective pressure boundary in the single-use bioreactor when looking to implement some kind of integrity test. And because the film and tubing on the single-use bioreactor bag are elastic, practical test methods must be established to attain useful results. Just like the visual inspection process mentioned earlier, collaboration with suppliers on this topic for recommendation and training is highly recommended. Accumulation of a large enough data set will allow periodic evaluation of the testing's effectiveness. And again, using a stainless steel bioreactor P&ID for reference, I've highlighted 4 addition lines and a sampling line. These steam-in-place lines include various combinations of manual and automated valves with weir type and radial diaphragms. The equivalent line types are present also in single-use bioreactors, but without the valves and especially automated valves. Instead, such lines on single-use bioreactors routinely use pinching devices, and the pictures shown here are examples of typical pinching devices used for single-use assemblies. Automation applied here to these devices on single-use bioreactors can improve process robustness by displaying procedural guidance, providing user prompts, and accepting acknowledgement that manual steps have been taken. To conclude this section, I thought it would be useful to compare agitator seal methods. The right-hand diagram is an excerpt from a stainless steel bioreactor P&ID showing a double mechanical seal. With it, there's a clean steam condensate seal lubrication subsystem. The component count and complexity are extensive, and besides the high level of maintenance associated with such a subsystem, mechanical seals still do represent a potential bioreactor contamination source. By comparison, the 3 images at the right show the elements that make up a representative single-use agitator and sealing mechanism. The magnetic drive head within the vessel opening is shown at the bottom. A cutaway of the impeller base placed on top of the drive head is shown in the middle picture. And up top, a complete impeller assembly with the bag weld flange highlighted in red. That is what actually makes the seal in this case. This is a much simpler design, and it eliminates the contamination risk posed by the other design. Trevor, back to you.
Thank you very much, Ken, for describing some of the operational considerations. And now I want to consider the future. The future bioprocessing facility will be a hybrid facility. It will most likely be a mixture of single-use and reusable platforms. For example, centrifugation still is a challenge for larger scale single-use, but where reusable equipment is needed, it will most probably be product dedicated to avoid the need for the cleaning validation product changeover. The ability to remove all the equipment from the room and fumigate will become standard practice, and clean room layouts will ensure unidirectional flow of people, materials, equipment, wherever possible. Such a facility will be capable of running any type of cell culture that you wish. The utopian view of bioprocessing is summed up in the next 2 slides. The progress that is being made with single-use systems means that we are moving away from uneconomical dedicated facilities. The meaning of multi-product facility is changing. At least 3 CMOs in the United States and many manufacturing companies are operating multiple single-use processes without the rigorous segregation that was required historically. One of these CMOs is running live virus, bacterial fermentation, and mammalian cell culture in the same suite on a campaign basis, and the regulatory authorities seem prepared to accept it. We're now seeing that the flexibility of single-use systems is driving demand. This in turn is driving improvements and is encouraging companies to design larger scale single-use bioreactors. The combination of larger single-use bioreactors and higher yielding cell lines means that it's now possible to go from bench scale to pilot scale to commercial scale in single-use systems in the same facility. If we can achieve this, we've saved an enormous amount of time and money during process and product development, and some companies have already started to realize these benefits. Now I'm just going to hand over to Ken for the last word.
Yeah, thank you, Trevor. Adding to Trevor's summary, we've covered a lot of material today. The benefits of single-use technology, including process flexibility, reduced cleaning needs, and the near elimination of contamination risk, are all reflected in better process economics. Sustainability, too, is well accepted now that single-use technologies have been broadly adopted. However, some questions still persist, like standardization of extractables and leachables testing, supported documentation completeness, and how effectively to manage critical quality attributes as part of a single-use life cycle management approach, and what remaining regulatory challenges remain, or will there be, as we push the technology further. However, we've presented some information we think you can use as you evaluate and implement single-use bioreactors and perhaps other single-use unit operations as well. Because of the importance of material management and single use, I wanted to be sure to highlight again how single-use technology increases the need for proper, adequate material management to support manufacturing, from the supply chain to the warehouse and from the warehouse to the bioreactor. And material management is not only about physical logistics. Effective single-use material management must include integrated information management as well. Implementing single-use bioreactors and single-use technology more generally requires a thorough operational analysis performed by a cross-functional team. Aligning equipment and consumable design spaces with appropriate user requirements goes a long way toward building a robust biomanufacturing operation. Single-use specific automation increases robustness even further. Site-specific equipment automation increases the availability of workflow and process data for use in continuous improvement activities. I'd like to thank the audience for their time and attention today, and thanks also to Trevor for teaming up on this presentation. For questions, answers, and discussion, Trevor and I would like to hand the webinar back to Sri. Sri?
Thank you very much, Ken and Trevor. Thank you very much for your presentation today. So I would just like to begin the question and answer session. Just as a reminder to our audience, if you have any questions for either Dr. Deeks or for Kenneth, then please send them to us via our questions tab at the bottom of your screen, and we will ask those questions in this section now. So first question, please can you elaborate on issues around the sensitivity of pressure decay integrity tests of single-use bioreactors?
Yeah, if I may, this is Ken. I think one of the things that's often overlooked in in the pressure testing, as Trevor pointed out, the bag itself, the film can't be pressurized. It's not really a pressure vessel like a stainless steel system. But the susceptibility to temperature differences, minor temperature fluctuations in the room where the testing takes place, I think is compounded really by the elasticity of the film material itself. I think it's important, and it's probably been discussed at conferences, that to really do pressure testing, pressure decay testing in single-use assemblies like bioreactor bags, there needs to be a fairly large data set to establish what's a positive result, what's a false positive result, and what's a negative result. And I think from the standpoint of industry standardization, it would be very interesting to see if this type of information could be anonymously pooled to be able to help all users benefit from a common body of knowledge.
Yeah. Just to add to that, Ken, one of the things that I've heard as a concern is the fact that you have got flexible film means that the pressure within the bag is subject to fluctuations of pressure and temperature in the room itself. Now, you should have a fairly constant temperature in a clean room, but pressure fluctuations can occur in clean rooms, particularly when people are coming in and out of the room all the time. And therefore, you need to be sure that you have control of those pressure fluctuations while you're doing the test itself. So that's an important consideration.
Great. Our next question, are particulates from the bags a quality concern? And if so, in what way can they impact quality?
Okay, I'll take that one. I think you have to look at particulates as in the same way as you look at leachables. Upstream, you're more concerned about how that might affect cell propagation and expression of your protein, because further downstream, you're going to filter out those particulates, unless, of course, you've got a product like a live virus that you can't filter, in which case, yes, the particulates are a concern, even upstream. Downstream, the bigger concern with particulates might be how they might affect your downstream processing. Will they affect the way in which your ultrafiltration membrane performs, or your filtration process, your filtration step itself? Or might they affect the chromatography resin in any way? Those are the concerns for particulates, and you just need to understand what they are. When it comes to the final drug product, you are sterile filtering the product, and therefore you shouldn't get any particulates anyway. All the particulates from your bags are going to be filtered out in the final fill finish, unless, of course, as I say, you've got a product that can't be sterile filtered. So sometimes I think there's a perceived problem more than a real problem. Do you have anything to add to that, Ken?
No, I think that was a good answer. I mean, I think for many cases for us, we see the issue being cleared up long before it becomes an issue downstream. It's usually not a consideration at the bioreactor level.
Great. So our next question, do you have examples of changing a process performed in SS to SU for a commercial product?
I personally don't. And I would think that there's probably not a lot of people who've actually done that for an established and registered process, because you really do have a lot of regulatory hurdles to get over. But Ken, have you got any examples that you know?
No, that's, I mean, your statement was really spot on. I mean, there has been discussion about it, and we know we've had a lot of customers, or let's say a lot, we've had a number of customers that have evaluated the possibility. They generally come to the conclusion that the switch is too costly at the point in the product's life cycle. So really the majority of what we see is processes transferred early in preclinical or early phase clinical, or even no transfer at all, and single use has been the entire technology used for a given process. I think that's becoming more and more prevalent. You know, I think probably the last 5 or 6 years, I mean, there's been a tremendous increase in the adoption rate, especially for larger scale clinical and commercial manufacturing operations.
Excellent. So our next question. Trevor pointed out process parameters can't be transferred directly from SS to SU and still achieve the same outcome. So 2 questions as part of that. Firstly to Trevor, what are the best practices you've seen PD groups take to achieve successful outcomes? And to Ken, what are suppliers doing to simplify the process and improve outcomes?
Yep, I'll take my part first. When you're doing design of experiments, you're not going to do them at scale because it would cost you a fortune and take you forever. You do them on scale down models, and it's a requirement. The regulators will expect you to demonstrate the scalability of your scale down models. You will have to validate your scale down model before you can do your design of experiments. So you have to show that what you do at small scale in the laboratory is reflective of what's happening in your GMP manufacturing scale. So the best practice is validate your scale down models before you do your design of experiments.
Yeah, I would concur with what Trevor said. I mean, I think for us as suppliers, we see 2 fundamental components. There's the biology or the science, and that really gets to the heart of what Trevor was saying, that as single-use equipment and single-use component suppliers, we are asked to provide information and support around process modeling, simulation, transfer of technology, things like that. And that's quite a bit different than the stainless steel days. The other component, the non-scientific, non-biologic component, is actually, the practical usage issues, the practical logistics around how do I adopt single-use technology? How do I use it effectively without putting the process or the components at risk? And as I mentioned, training is a big element of that. We see lots of dividends being paid back in terms of continuity and lack of failures by doing proper training of resources at the customer site. I mean, one of the simplest things is just, how do you deal with a single-use box, single-use containing box in the warehouse, and how do you stage it for manufacturing? And that's why I really emphasize those 2 elements today. You sometimes don't think about what you're wearing. If the warehouse guy has keys on a tool belt, or the forklift is buzzing around, but those types of things do eventually end up causing damage, and some of that damage does make it through into the manufacturing space.
Thanks. So our next question. So currently, these processes are limited to about 2,000 litres. So what scale do you both see being achieved in the future, and what timescales may apply?
Interesting.
Well, there is one company that's got a 3,000 litre bag bioreactor out there, a single-use 3,000 litre bag. I don't know how successful it is. I don't have any personal experience, but I mean, I think that the better way to scale up these processes is not to keep thinking about bigger and bigger bioreactors, but to think about more of them and then pooling. I mean, that's part of what we discussed, isn't it?
Yeah, absolutely. Yeah, I think that, you know, if you look at a little further back in the history of the bioreactor space and biomanufacturing space, you know, around the year 2000, 20,000 liter stainless steel bioreactors seemed to be pretty much the norm, with, you know, several companies buying 6-packs of these things for huge volumes. And there were lots of reasons behind that. But there are also reasons not to take that approach as well. I think for single-use systems, you know, it's been a convergence of improved process performance, the industrialization of CHO cells, and a lot of other scientific and biological process knowledge has been able to improve the productivity of bioreactors in general. So, you know, I think 2,000 liters was a pretty common evolution in the size to achieve the proper amount of production volume or production capacity based on the relative or comparative risk associated with putting a lot of fluid in a big plastic bag. But, you know, the marketplace may change. I mean, you know, there's a lot of discussion around process intensification, a lot of discussion around continuous bioprocessing, and that certainly doesn't point to larger bioreactors. It points more to smaller bioreactors that are more efficient, getting more product per unit time from smaller scale. So, you know, we'll have to see how that evolves. You know, there's certainly some examples of larger scale bioreactors than 2,000 liters, as Trevor mentioned, but I don't think at this point that's the norm or is expected to replace where we're currently heading in the industry.
I think just on the business of the cell line as well, if the vendors of the cell lines are to be believed, you can get the same yield out of 2,000 liter bioreactor now that you used to be able to get out of a 20,000 liter bioreactor maybe 10 or 15 years ago. I don't know to what extent that's true, but certainly the yields are much better than they used to be.
Great. Thanks very much. So our next question, has training reduced bag failures in practice?
Yeah, I mean, we see evidence of that all the time. It's particularly true with organizations that are either new to single-use bioreactors as a more complex single-use assembly, or as they transition through different groups. So process development typically has scientists and engineers who understand the technology, who work with it a little bit more closely. As you transition that into a manufacturing process, some of the qualifications of the operators are not the same level, so they require some additional or supplemental information making that transfer. But yeah, we do see improvements. We do see success rates increase, bag failure rates go down as a result of handling practices.
And the next question, just quickly, have you seen processes that could not be transferred from SS to SU?
I haven't personally, but I've spoken to people who said, colleagues who said yes, they had a cell line and it wouldn't run because the cells adhered to the surface layer, the inner surface of the bag, and they couldn't get them to do anything. I've also heard people who said that the protein itself will adhere to the bag, and therefore they lose a lot of yield as a result.
Yeah, that's true. I mean, I've heard similar examples. Those are good ones. You know, I think that besides those, there are other processes where either the productivity, getting back to the previous question, you know, the productivity from the cells is not matched to the scale or the volume of the single-use bioreactor, even if you scale it out. I think the ability to address the variety of different cell culture processes has expanded over the years, and I think it's going to continue to expand as improvements are made in the technology. But I think, you know, if I use single-use fermentation as an example, you know, that's available now in single use. I mean, you go back 5 years ago, probably nobody thought that was possible, that the bag would be able to handle that type of process requirement and that type of physical stress. But I think as the industry has adapted, all the components, including the films and other plastics, as well as the capabilities of the bioreactor hardware, have increased to address the requirements that customers have had.
Okay, well, thank you very much, gentlemen. Unfortunately, that's all the time we have. Any questions that haven't been addressed here live will be answered via email. I'd like to take this opportunity to thank our presenters, Dr. Trevor Deeks and Kenneth Clapp, for sharing their knowledge with us. Audiences can view this webinar on demand by visiting biopharma-asia.com/webinars. Thank you very much, everyone.
Thank you. Have a good day.
Thank you.